Welcome to the last video in the unit on bacterial genetics. In this video, we're going to look at mechanisms of horizontal gene transfer, which are ways that bacteria are able to increase the variation since they are not sexually reproducing organisms. Let's take a look at these mechanisms. Horizontal gene transfer is defined as the transfer of genetic material between different bacterial cells. This is different than vertical transmission, which is when genetics are passed from one generation to the next, such as to daughter cells during binary fission. Instead, horizontal transfer is the transfer between cells that were not related, and in fact are sometimes not even the same species. Horizontal gene transfer is another way that asexual bacteria can increase their genetic diversity by sharing genetics with each other. There are three main methods of horizontal gene transfer that we're going to look at. The first is transformation, which involves taking up naked DNA into the cell. Transduction, which is using phage to transfer DNA between bacterial cells, and conjugation, which requires cell-to-cell contact for transfer along a sex pelus. It's important to remember that bacterial DNA is typically circular form, both for the genome and for plasmids. Thus, the cells are not used to linear forms of DNA and will degrade them if they enter the cell. This means that any linear piece of DNA that's transferred in must first integrate into a piece of DNA that's already circular and in the cell. This integration is called recombination. So for example, if a linear piece of DNA came into the cell, it would have to recombine into the bacterial chromosome. That happens by inserting and swapping places with a piece of DNA that's already there. Usually parts of these regions are going to have to be homologous, where they have similar sequences. And so after recombination, the genome now will have this new piece of DNA inserted where the old piece had been. That process again is called recombination. Transformation is a process where bacterial cells are able to bind free-floating DNA in their environment and take it into the cell. Often this free-floating DNA is parts of the genomes of other bacterial cells that have died and begun to break down, releasing their DNA into the environment. Species that are able to do this naturally are called competent cells. And they're competent because they have a receptor on their cell surface that's able to bind to the DNA, which then allows the DNA to travel through a transport protein into the cytoplasm. If that DNA is a linear piece of DNA, it's going to have to undergo recombination to become part of the genome itself. This allows it to become a permanent part of the cell's DNA. This can also happen with plasmid DNA. If transformation with plasmid DNA happens, then the plasmid's already circular, so it doesn't need to go through recombination. It can just hang out in the cytoplasm and function there as a plasmid. In the lab, we're able to take advantage of transformation because you can make cells artificially competent by mixing them with certain chemicals. This then allows them to take up DNA through transformation, even if they wouldn't have been able to do so naturally in their environment. We do this to genetically modify bacteria. So for example, the E. coli that produces most of our human insulin, that human insulin gene was put on a plasmid, and that plasmid was inserted into the E. coli through transformation. Once it's in its cytoplasm, it's going to transcribe and translate the insulin gene, producing the insulin protein for us to use. While transformation allows bacteria to pick up DNA from their environment, conjugation requires cell-to-cell contact, and it only allows the transfer of a specific plasmid. The plasmid involved in conjugation is called the F plasmid, and it contains the genes needed to make the conjugation pelus and transfer the DNA. This means that only F-positive cells can initiate conjugation because those are the cells that contain the F plasmid. F-positive cells always initiate conjugation with an F-negative cell that does not contain an F plasmid. When conjugation is initiated, the sex pelus forms, connecting the two bacterial cells together. One strand of the F plasmid then unwinds from the plasmid and is transferred through the F pelus, where it will then recircularize in the F- cell. Once both cells have a single strand copy, it's able to be used as a template to replicate the second strand, leaving two F-positive cells at the end that have the fully formed double-stranded F plasmid. The transfer causes the F-negative cell to become F-positive, which means it's now able to form a sex pelus and initiate conjugation to other cells. Besides the genes for conjugation, these F plasmids can also contain genes such as antibiotic resistance that may be beneficial to the bacteria and also get transferred to the new cell through conjugation. Interestingly, sometimes the F plasmid will integrate randomly into the bacterial chromosome. When this happens, all of the genes for transfer are now part of the bacterial chromosome instead of on a separate piece of DNA. These are called HFR cells for high frequency of recombination. They are named this way because recombination is going to be required in the F negative cell, unlike when the F plasmid was a separate piece of DNA. So now when conjugation is initiated to an F minus cell, it's not just the F plasmid that's transferred, but also part of the chromosome itself. The sex pelus is not stable enough to pass the entire chromosome through, however, so it's only going to pass a part of it. And so we'll get part of the F plasmid that's attached to part of the chromosome, but that's now a linear piece of DNA. Since the entire thing doesn't come through, it's unable to reform the circle. Being a linear piece of DNA means that this is now going to have to undergo recombination to become part of the chromosome if it's going to remain permanently in the cell. HFR cells are able to transfer not just the F plasmid genes, but also whatever genes were on the chromosome that got pulled through that sex pelus at the same time. However, because part of that F plasmid was still on the chromosome at the other end, it never fully makes it into the new cell, and so those F negative cells remain F negative. They don't get a fully functional F plasmid after this transfer. Alternatively, the integrated F plasmid will sometimes pop back out and reform a plasmid. If this happens, it will often pick up a little bit of extra DNA from the chromosome itself, gaining a new gene or two that will now be involved in transfer. Cells that contain these slightly larger F plasmids with little pieces of the chromosome are called F prime cells. The last way to transfer DNA horizontally between two cells is transduction. However, in transduction, the bacteria don't actually have any mechanisms that they're using. Instead, they just accidentally end up with some new DNA because of a mistake by the bacteriophage. This method is an accident, not a physical method the bacteria are controlling. Transduction occurs when there's a mistake during the bacteriophage assembly step. Instead of packaging viral DNA, the new virus instead packages up bacterial DNA. This happens because often the phage caused the breakdown of the bacterial genome into separate little pieces, so that the bacteria cell can't replicate or transcribe or translate its own genome and instead is functioning solely on the viral genomes. When this happens, sometimes that bacterial DNA might end up inside one of the phages during assembly. When this phage is released, it's now carrying bacterial DNA, and when it goes to infect another cell, it's going to insert the bacterial DNA instead of the phage genome. This means that this cell here got lucky. It didn't get infected by a bacteriophage. Instead, it gained a new piece of DNA, and since that DNA is linear, it's going to have to recombine into the genome to become permanently part of that bacteria's DNA. There are two types of transduction. Generalized transduction is when any random piece of the genome is transferred, while specialized transduction is unique to lysogenic phage. These phage integrate into the genome at a specific point, and the incorporation of bacterial DNA comes when the phage genome comes out of the chromosome to switch back to the lytic cycle. When this happens, it accidentally can take a piece of the bacterial DNA that was right next to it, but this is always the same piece of DNA, so in specialized transduction, only one specific region of DNA is transferred every single time. It's not random like generalized transduction is. We've discussed recombination as a way for linear DNA to become incorporated into the bacterial chromosome or into bacterial plasmids. But another way that DNA can become integrated after it's transferred is if it contains a transposon. Transposons are thought of as jumping genes. They're able to be cut out of one piece of DNA and inserted into a different piece of DNA, kind of like cut and paste in a Word document. This happens because of an enzyme called transposases. These transposons are specific sequences in the DNA that have an inverted repeat at each end, so an inverted sequence that says this is the end of the transposon. And in between those is the gene for the transposase enzyme. That enzyme is what recognizes the transposon, cuts it out of one piece of DNA, and inserts it into another piece of DNA. In between those inverted repeats, you can also have other genes, like genes for antibiotic resistance, and so if that transposon jumps into a new piece of DNA, it brings those other genes along with it. Transposons are not unique to bacteria the way the other horizontal gene transfer mechanisms are that we've been talking about. In fact, they're found across all domains, and there's actually remnants of thousands of them that are found in the human genome. This highlights the fact that they really contribute to increased genetic variation in all different populations.